Hello.
Good evening, madam.
Uh, Good evening, Jubin. I will start today. Before starting, I will ask whether there is any doubt for you, like in the previous lectures, or any question are there?
No, madam, not right now. I have not gone through all the classes. I mean, this week's. I'm just
Okay,
going through only. So far, no doubts, madam.
okay. So last week I have been teaching like about innate immunity and what are the different PRRs or pathogen recognition receptors which are involved in sensing the pathogens. I'll continue that because it is a big chapter. So today I will talk about the different uh, peptides or the different kinds of interactions between innate and adaptive immunity, and also uh, like about the cytokines in brief. So first, I will again begin by uh, discussing about what are the different types of innate immune proteins which are induced by PRR signaling. So as we can see, like there are different kinds of proteins by innate immune signaling. So there are antimicrobial peptides as well as proteins like defensin cathelicidins, interferon alpha and beta. These are antimicrobial compounds which are produced by different kinds of cells like epithelial cells, neutrophils, natural killer cells, virus infected cell, macrophages. They act on different kinds of cells, other types of cells, like it can affect the same cells or it can go and affect the other cells which are infected by this pathogen. So their role are like they can inhibit viral replication, they can activate degranulation, they act as chemoattractant, they regulate the different activity of the other bacteria and all. So apart from these antimicrobial factors, there are certain cytokines which are small molecules, which are small proteins, which are produced by this monocyte macrophage dendritic cells. There are different kinds of cytokines like IL-1, IL-6, TNF-alpha, IL-12, IL-10, IL-18, GM-CSF. So uh, these cytokines will have different effects. Some are pro-inflammatory cytokines, some are anti-inflammatory cytokine. Pro-inflammatory means it helps in inflammation, and anti-inflammatory means it reduces inflammation. Example of one anti-inflammatory cytokine is IL-10, which are produced by this macrophage dendritic cell mast cells, and others are mostly pro-inflammatory, like IL-1 here in this table, TNF-alpha, IL-6. So they induce inflammation. So apart from cytokines, there are other molecules which are known as chemokines, which are chemoattractants and are produced by macrophage dendritic cells, vascular endothelial cells, and they basically what they do is they chemoattract other cells to the infection site. Like whenever there is an infection in the cells, they attract other cells like neutrophils, basophils, etc. to come into that site of infection. So these are the different kinds of innate immunity proteins which are induced by PRR signaling. So I will go one by one. What are the different kinds of innate immune proteins? First, I will come to antimicrobial peptides. Uh, like antimicrobial peptides include type 1 interferons, as we have already seen here, interferons alpha, beta. So, what are the different kinds of major antimicrobial proteins? They are the major representatives are the IFN alpha and beta, the type 1 interferons, and they are directly transcriptionally induced by these pathogen recognition receptors. So, whenever there is an infection with virus, many cells can induce the production of this IFN alpha or IFN beta following binding of cytosolic viral PAMs, usually nucleic acids, to intracellular PRR such as uh, ALRs, RLR, and C gas. So many uninfected cells are there which express the surface TLRs or the toll-like receptors that can recognize different kinds of PAMs or pathogen associated molecular patterns, which I have already discussed last week, and they can internalize this virus without being affected, allowing these endosomal TLRs to recognize the viral components. So these kinds of uninfected cells also will can get affected by this type 1 interferons and can further induce the production of more and more interferons. So there are other specialized type of cells with a dendritic cell. There are different types of dendritic cells. One type is the plasmacytoid dendritic cell, and because of its shape only, it is named like that, plasmacytoid, and it is particularly effective in producing this type 1 interferon. And what these PDCs or plasmacytoid dendritic cells do, they endocytose the virus that has bound to various cell surface proteins, such as like one example is like HIV receptor is there, DC sign receptor, which is again a receptor for both HIV and dengue virus. And so these PDCs endocytose these viruses that has bound to various cell proteins and help in the help in the production of this interferon. And apart from this, there are TLR7 and 9 in the endosomes which are present, which are activated by the viral PAMs, which leads to the activation of IRF or interferon regulatory factor and type 1 interferon production. So this type 1 interferon production is like very common whenever there is a pathogen entry, and many cells actually produce this type 1 interferons. So this is the signaling uh, process by which these type 1 interferons are produced. So here we can see two interferon dimers are there, like interferons alpha and interferon beta. These are the major two IFN like interferon one IFN ones which can bind and dimerize the interferon alpha receptor. Like here we can see there are two receptors here. Interferon IFNAR1, IFNAR2. These are the two interferon alpha receptor which are present, and for the starting of the signaling, these two needs to dimerize. So whenever there is a binding of the ligand to the receptor, these two dimerize, and then it activates and recruits other protein kinases like JAK and tyrosine kinase. So these are tyrosine kinases which are required for activation of the signaling. So whenever there is a binding of the ligand to the receptor, there is dimerization and there is phosphorylation of these JAK kinases, which further phosphorylates other proteins which are STAT1 and STAT2. So these STAT1 and STAT2 then dimerize together and enter the nucleus to further stimulate the expression of proteins that activate antiviral effects. So uh, these signaling is very much important. This phosphorylation is very much important to activate the downstream signaling. If we by somehow this phosphorylation is blocked, then this activation of signaling will be stopped. So this dimerize the stats that stat1 and stat2 is dimerizing and entering into the nucleus, and it is leading to the transcription of different interferon stimulated genes. So there are certain genes which are known as ISGs or interferon stimulated genes which are being transcribed in response to any pathogen response in pathogen entry. So these interferon stimulated genes, there are four major interferon stimulated genes. Apart from that, many are there and many new are coming up. So these are the most important or four ISG which are known for inhibiting viral replication, and they are for one is protein kinase R, another is OAS or 2,5 oligonucleotide A synthesis, and another is MX group protein, other is IFIT interferon induced proteins with tetracopeptide repeats. So first is protein kinase R, which binds and is activated by double-stranded RNA. It then blocks viral and cellular protein synthesis by inhibiting the translation initiation factor EIF2 alpha. So what it does, it is bound by double-stranded RNA viruses. So whenever it is binding to this RNA viruses, here you can see double-stranded RNA viruses is coming and it is binding to this protein kinase R. It can recognize this double-stranded RNA. And what it does, it inhibits one translation initiation factor, EIF2 alpha. So uh, this leads to the inhibition of translation. Like as a whole, it will block the translation process, after which there will be no synthesis of protein. It will block both virus because it cannot only target the viral protein replication, because as a whole this EIF2 alpha is needed for our protein synthesis also. So it will block virus as well as cellular protein synthesis by inhibiting this EIF2 alpha. So the virus, whenever the virus is cleared uh, from our body or from the cells, it will again, this inhibition will again like get back to the reverse form. So another one is this oligo A, 2,5 oligo A synthetase. This is a OAS. We call it as OAS. What it does, it is actually nucleotidyl transferase, which is related to C gas. After its expression is induced by IFN, OAS binds cytosolic DS RNA, which activates it to generate 2,5 2,5 oligoadenylate from ATP. Oligoadenylate then binds RNA cell and induces it to degrade viral RNA. So here 2,5 oligosynthetase is synthesizing this oligoadenylate 2,5 oligoadenylate, which is recognizing this double-stranded RNA, uh, which is activating a ribonuclease RNase L, and that is degrading this mRNA of the virus as well as cellular mRNA. So the virus, what they do is they are dependent on the host machinery only. They will depend on our translation factors. They will depend on our enzymes, even different, even their which is needed to transcribe their genome. So they will depend on the host machinery uh, so much that whenever there is a viral entry, then this proteins will be synthesized. These different kinds of ISGs will be synthesized, which will actually stop these major processes like translation, or it will degrade the mRNA so that the virus cannot use our machinery and synthesize their proteins, or they cannot transcribe. So Another group is the MX group of proteins which inhibit both the transcription of viral gene into mRNAs and the assembly of virus particles. So these MX proteins will convert GTP to GDP plus P, and this they will self-assemble together. You can see these are the complex formed by the MX proteins, and they will form a ring-like structure and will inhibit viral transcription as well as assembly. So another is the IFIT proteins, which will bind to double-stranded RNA, blocking viral RNA translation. Several IFITs also bind and inactivate the EIF3 translation initiation factor. So these IFIT proteins bind to viral RNA as well as EIF3 and will block the translation of the viral protein. These are the four major groups of um ISGs or interferon stimulated genes, and all these are produced downstream of this interferon signaling. So apart from these antimicrobial peptides, I told that there are certain molecules which are cytokines. These cytokines are small molecules, and they are the different types of proteins which are transcriptionally induced by PRR activation. Uh, they are not directly antimicrobial, but they will activate and regulate a wide variety of cells and tissues involved in innate inflammatory as well as adaptive responses, and they will function as the protein hormones of the immune system. Means they can be produced in response to one stimuli, and they can travel long distance like hormones and can act on a different types of cellular targets. And most of the three important cytokines are IL-1, TNF alpha, and IL-6, and these are all pro-inflammatory cytokines. They will act locally on the blood vessels to increase the vascular permeability, and also an other cells, including lymphocytes, to recruit and activate them at sites of infection. So, as I already told, like these cytokines are very much important, and they will act from one distance to another. They can act in the like it is if it is produced by one cell, it can act on the same cell, or it can act on a different cell which is very much far away from that particular type of cell. And it also many types of adaptive immune response are being controlled by the cytokines. Like it can generate like the one from the innate cells. If cytokines are produced, it can go and activate the adaptive response also. So cytokines are very much important, and there are two kinds. One is pro-inflammatory cytokine and another is anti-inflammatory cytokines. And most are, as I told in this table, most are pro-inflammatory cytokines. This IL-1, IL-6, TNF alpha, and one is anti-inflammatory, that is IL-10. So this is a signaling through this TNF alpha, which is again a pro-inflammatory cytokine. Okay, I'm checking. So these cytokines are very much important for defense against these pathogens, all kinds of pathogens, especially virus infection. So this is one of the important cytokines, that is TNF or tumor necrosis factor, and you can see that this is a signaling process by which this TNF alpha acts. And here you can see like this is a plasma membrane and this is a TNF trimer which is forming. Like mm, this receptor homodimerization,
Madam,
heterodimerization. Yes, yes, hello.
excuse me, madam. Sorry to interrupt. The slides are not.
Not
Uh,
changing.
yeah. Now I can see the slide, madam. Meaning like now slide number 7. Earlier it was just showing 6. That is why.
Here you can see
Yes.
six,
Uh, now it's okay. Yeah.
six. You can see no changing. I
Yes.
am changing the slide. You can see no.
Yes, madam. Yes. Thank
Okay,
you.
okay, okay. This TNF trimer, this TNF or TNF alpha acts as a trimer. So this homodimerization or heterodimerization of receptor is very much important, because if you mutate one and if two are coming, then it won't activate the signaling. So it should be, if TNF is signaling through a trimer, it should be a trimer. It cannot activate the signaling if it is a dimer. So this binding of TNF, which is a trimer to the receptor, like TNF receptor is this. So this three will come together and there will be a like binding of the ligand. Ligand here is TNF alpha, which will bind to this receptor, and it is having different kinds of domain. Like this is the death domain, which is present inside the cytosol. Uh, and this here you can see that this is a dead domain, and it recruits different kinds of adapters, different kinds of proteins it recruits, which again transmit the signaling. So binding of this TNF to the receptor will activate this death domain, which will recruit different kinds of adapters here. So these adapters, for example here trad, then will bind to other proteins. This is just a signaling, and there are different kinds of proteins which are coming, and it can lead to two different types of signaling. One is it can activate the NF kappa B pathway, or it can activate the MAP kinase pathway, and there will be activation and survival of innate and inflammatory genes. Then this is one, like it will, there are two functions of this TNF alpha. One is like it can activate and uh, many type of different types of inflammatory genes, and it can help in the survival of the cells. And another is it can induce apoptosis. Either it can help its survival, or if it is seen that there is no chance that a cell can survive and it has to die, it will initiate apoptosis. Apoptosis is programmed like induced cell death. Like whenever there is some cells are not necessary, then those will go and undergo apoptosis in our body. So here you can see these are, this apoptosis signaling is done by different other kinds of adapters. Like here you can see CIAP, Lupax, these are different kinds of proteins which are not present. So it depends on the situation at that time which pathway it should activate, whether it should go to the survival pathway or whether it should go to the apoptosis pathway. So here, this is just a signaling. No need to just remember or memorize. This just showed that depending on the type of the situation in the cell, these cytokines or the antimicrobial peptides can act differently. So apart from antimicrobial peptides and cytokines, there are other proteins like this is chemokines. There are chemokines, or as I already told, they are chemoattractants. Like they will recruit cells within and out of the tissues. Like as I told, like they will help other cells to come to the site of infection. So chemoattractants means agents that induce cells to move toward higher concentration of the agent. Like whenever there is that particular chemoattractant is present, it will induce the cells to come to that site. So some chemokines are responsible for constitutive migration of white blood cells throughout the body. Like I told, the first cell to come at the site of infection are the neutrophils. Whenever there is any breach or pathogen coming, so neutrophils comes first in that section. So what these chemokines are secreted from that section, and they will help in the migration of these neutrophils to that site. So other chemokines are produced in response to PRR activation. They have key roles in the early stages of immune and inflammatory responses in that they attract cells that contribute to both clearing the infection or damage and to amplifying the response. They can either contribute in clearing the infection or they can help in amplifying the response, the immune response. So, first chemokine which was cloned was IL-8. So, IL-8 or CXCL8 is produced in response to activation by the pathogen associated molecular patterns, damage associated molecular patterns, or some cytokines of a variety of cells at sites of infection or tissue damage, including macrophage, dendritic cell, epithelial cells, and vascular endothelial cells. So, one of the key role of this IL-8 is in the initial stage of infection or tissue damage. Like it serves as a chemoattractant for neutrophils, as I told. Like they will attract these neutrophils and they will recruit them from the blood to the sites of infection. So there are, apart from these chemokines, there are certain enzymes, INOS and COX-2. They are produced in response to PRR activated signaling pathway. INOS is inducible nitric oxide synthase and cyclooxygenase 2 is COX-2. They have the role in generation of antimicrobial and pro-inflammatory mediators. The INOS enzyme catalyzes one important step, like it is already known as inducible nitric oxide synthase. That means it is induced and it is a nitric oxide synthase. It will help in the formation of nitric oxide, which will kill the phagocytose microbes, and it will be, I will show like how it will kill. So COX-2 whose synthesis is induced by PRR activation in monocyte, macrophage, neutrophil, and mast cell is key to converting the lipid intermediate arachidonic acid to prostaglandin potent for pro-inflammatory mediators. So COX-2 is a another enzyme which is produced by PRR, and it is induced by the PRR or the pathogen recognition receptors in monocytes, macrophage, and neutrophil or mast cells, and they will like convert the lipid arachidonic acid to prostaglandin, and prostaglandins are known to be pro-inflammatory mediators. So these are the basically innate immune proteins, these antimicrobial peptides, these enzymes, these chemokines, these cytokines, which are helping in producing the response and amplifying the response whenever there is any pathogen entry. So apart from that, we know there are a process known as phagocytosis. It is a important mechanism for eliminating pathogens. Like they make up an important line of defense against pathogens that have penetrated the epithelial cell barriers. So phagocytic cells include, as I already have told in I think second class, like there are different types of phagocytic cells, and they are monocytes, macrophage, neutrophils, dendritic cells. All these are innate immune cells, and they will clear up the infection by a process known as phagocytosis. So monocyte in the blood, macrophages, neutrophils, dendritic cells, and in tissues are different types of cells which carry out phagocytosis. That is, phagocytosis means the cellular uptake and eating and destruction of the particulate materials greater than 0.5 microns in size, such as bacteria. So it will eat up and it will take up, there will be protrusions from the cells, and they will take up the any kind bacteria or other material which is greater than zero point 0.5 microns, and they will destroy that particulate matter. So most tissue contain resident population of macrophages that function as sentinels for the innate immune system. These macrophages are derived from embryonic precursors that seeded the tissue during early development. So our tissue always contains the macrophages, and different kinds of macrophages are present in our different organs, and they are given different name. Those are the tissue macrophages, and they will function, they are mostly important for this phagocytosis process, and they are present from the when we were at the embryonic stage only during early development. And another type, as I already told, neutrophil, they are recruited from the blood to the sites of infection. I told chemoattractants will attract these neutrophils, and they will have, neutrophils means they will have some granules inside that will contain this antimicrobial proteins peptide, and will be, it will fuse with phagosomes, and dendritic cells also can bind and phagocytose microbes. So these are the main different types of cells which are uh, helping in the phagocytosis process. uh, so any kind of question is there or any doubt is there? Okay, okay, just 2 minutes. So this is the, you can see this yellow is the macrophage, and this red is the bacteria. So it is surrounding the bacteria, and it will internalize and eat up the bacteria. So this process, here is the process how this phagocytosis takes place. Here you can see There are certain structures in vagination which we call as pseudopodia. This is a macrophage, and whenever there is an entry of this bacteria, it will bind to different kinds of receptors. There is a pathogen recognition receptor which are present on the surface of this macrophage, and they will interact. That bacteria will have certain antigens which will interact with these receptors, not antigens, some ligands, and there will be a protrusion of the cell surrounding the bacteria. Then what they will do? They will uh, take up the bacteria by forming this protrusion, and it will go into the this phagosome, that is phagocytosis. Since the process is phagocytosis, instead of endosome we are calling it as phagosome. So this phagosome is having a pH of 6.2. So this phagosome then then interacts with the lysosome. Here you can see lysosome. We all know lysosomes are certain organelles present in the cell which will produce certain enzymes and will degrade the whatever is entering into the lysosome. So this phagosome enters this lysosome and it will form phagolysosome. This is a phagolysosome. So this lysosome will uh, secrete these enzymes, and you can see the pH suddenly drops whenever it fuses, because this lysosome enzymes will act only at the acidic pH. So here the bacteria then gets digested and killed by these enzymes of the lysosome, and then these digested products are then released from the cells. So uh, this is the process by which this phagocytosis uh, takes place, and they will uh, like first is a process where it will form a pseudopodia. It will internalize the microbe, then it will digest the microbe, and then will release the digested products which will be released from the cell. So there are different types of phagocytic receptors which are present, as I already told. In I showed these are the receptors which are present, like the phagocyte like phagocytic receptors on the surface of these cells like macrophages, monocytes, etc. And many of them are PRRs or pathogen recognition receptors, and they will directly recognize the PAMs or the pathogen associated molecular patterns present on the surface of the microbes, and like the cell walls of the bacteria or the fungi. But all PRRs are not known to induce phagocytosis. For example, TLRs or toll-like receptors are a major class of PRRs that do not induce phagocytosis. So whatever surface receptors is present on the macrophage are not all are PRRs, but many are PRRs, and those PRRs will induce phagocytosis. But one of the major class is TLR, which will not induce this phagocytosis. Uh, so this most PAMs or the pathogen-associated molecular patterns that induce phagocytosis are cell wall components, including complex carbohydrates such as mannans and beta-glucans, LPS, other lipid-containing molecules, peptidoglycans, surface protein. So these PAMs, as I already told, will interact with the PRRs like the LPS present on the bacteria, such as some lipid-containing molecules such as peptidoglycan or mannans, beta-glucans, and other fungal components. It can interact and can activate the phagocytosis process. So, activation of phagocytosis can also occur indirectly by phagocyte recognition of soluble proteins that have bound to microbial surfaces, thus enhancing phagocytosis. And this process is known as opsonization. So, opsonization, it came from a Greek word which means to make tasty. So these phagocytes, or the cells which are involved in the process of phagocytosis, are known as phagocytes, and they will recognize some proteins which are bound to these microbial surfaces and will enhance this phagocytosis. And that process is known as opsonization. And many phagocytosis enhancing proteins are known as opsonins. They also bind to conserve repeating components on the surface of microbes such as carbohydrate structures, lipopolysaccharides, viral proteins. Hence, they are sometimes referred to as soluble pattern recognition proteins. So there are certain proteins which can also induce this phagocytosis, and they are known as opsonins. And these they will bind to the microbial surfaces and by a process known as opsonization. And once they are bound to the microbe surfaces, they are recognized by membrane opsonin receptors on phagocytes, thus activating phagocytosis. So here you can see these are the different kinds of human receptors that trigger phagocytosis. For example, I already told not all will be pattern recognition receptor, but there will be other receptors also. On a C-type lectin receptors, example are mannose receptor, dectin-1 DC sign receptors, which is a receptor for both dengue as well as HIV. Then this mannose receptor will interact with the mannans present in bacteria, fungi, and parasites, and this dectin-1 will interact with beta glucans which are present in fungi or some bacteria. And this DC sign will be interacting with the mannans which are present in bacteria, fungi, and parasites. Then there are other receptors like scavenger receptors, SRA, SRB. They will recognize the lipopolysaccharide LPS, lipoteichoic acid from the bacteria. And this SRB also similarly as well as beta glucans from fungi. So these will interact, these microbial ligands will interact with these receptors on the surface of these phagocytes. There are also opsonin receptors like collagen domain receptor, complement receptor, immunoglobulin Fc receptors. So collagen domain receptor is calreticulin, that is collectin SBA. These are the different types of ligands which will be activate this receptor, and there are complement components and fragments which will activate this complement receptor, and there are immunoglobulin receptors which will be activated by these antibodies bound to antigen, and this IgG will activate the this Fc gamma receptors present. So this will opsonize, and this will lead to the opsonization process and thus enhance phagocytosis. These are the different kinds of receptors that will trigger phagocytosis. So here you can see, as I already told, this mannose receptor is there, and it will react like it will interact with this mannans. So mannose binding lectin, which is a lectin is a complex of these multiple polypeptide chains, and each will contain N-terminal cysteine-rich region followed by a collagen-like region and a recognition domain. So this is a mannose binding lectin. Uh, these are the different like a collectin or the different receptors which will bind to the mannose. These mannose receptors. This will form a complex of polypeptide chain and interact with these mannose which are present in different bacteria, fungi, and they will there are different kinds of serine proteases, MASP. So you can see these are MASP, which will become active after the recognition domains bind to the specific residues on the surface of the pathogen. And then it can activate the complement pathway. So complement pathway I will teach next day what is component, complement. So complement also will when it will get activated and it will bind and it will form a complex with the pathogen, and it can trigger phagocytosis or opsonization also. So here you can see how it is forming a complex and recognizing this surface molecules of the bacteria, and then it's triggering the complement pathway. So there are other processes also which can kill the phagocytosed microbes. Uh, like I already told, like this phagocytosis occurs, and then they will digest the microbe and they will release this digested parts out of the cell. So these are the different types of processes or the antimicrobial species are generated and nitrogen. Like here you can see many reactive oxygen species are produced like superoxide anion, hydroxyl radical, hydrogen peroxide, hypochlorous acid, and here some reactive nitrogen species also are produced like nitric oxide, nitrogen dioxide, peroxynitride, and these all will also has the antimicrobial activity. So they will directly kill the phagocytosed microbes. So these are produced inside the cells, like in the cytoplasm of neutrophil, macrophage, dendritic cells. Several enzymes, including NADPH oxidase, transfer molecular oxygen into highly reactive ROS that have antimicrobial activity. And another is the this nitrogen species, like the superoxide anion can interact with the reactive nitrogen species RNS. Here you can see the superoxide anion of the ROS pathway will interact with this nitric oxide and will form a peroxynitride, and uh, this all species will then lead to the killing of the phagocytous microbes. So there are different kinds of uh, signaling as well as different kind of production of this uh, antimicrobial species are generated inside the cells which will kill these phagocytous microbes. So apart from these molecules also there are other and this phagocytosis also there are other methods by which we can eliminate the pathogen. So there is a process which is known as netosis, and there are nets or neutrophil extracellular traps which will help in the elimination of this pathogen. Here you can see these are the pathogens, and this is the neutrophil extracellular traps. It will trap the pathogen and will kill the pathogen from the cells. Here also you can see a microscopic image where you can see a net is produced by an activated human neutrophil dying due to netosis and four non-activated neutrophils. These are the activated, this one is the activated one neutrophil, and these are the four non-activated uh, neutrophils, and it is like netosis is the process by which these neutrophils are dying, and it is forming a complex and it is forming a net kind of, and it will lead to the pathogen elimination. So these neutrophils are coming in the site of infection, and it is also having this PRRs on its surface or the pathogen recognition receptors, and there will be binding of the ligands from the microbes to these recognition receptors, and that will activate the signaling as I already showed. Like there will be formation of this species, and there will be NADPH oxidase which is induced, and there will be breakdown of the intracellular membranes. Like here you can see there is breakdown of these intracellular membranes will start, and this neutrophil already contains granules which will contain some enzymes like elastase and myeloperoxidase, and which will enter the nucleus and modify the histones, and then which will lead to the decondensation of the chromatin. Like this superoxide radicals will be formed. This will decondense the chromatin. There will be release of there will be modification of the histones, and there will be release of the nuclear components as well. And then ultimately what happens? The plasma membrane will rupture, cytoplasm and the nucleoplasm are expelled and forming these nets, these structures. They will form like a trap kind of, or the net kind of, and the cell will die from netosis. So this process also helps in the elimination of pathogen, and it is one of the important process which is happening inside the cell, where the neutrophils are actually killing the pathogen by directly inducing cell death. That the affected neutrophils will die by formation of these net structures. So this, apart from this netosis, there are other processes. As I already told, one is apoptosis, which is program induced cell death, and this is like another is process is pyroptosis. Actually, these are the different types of regulated cell death. Regulated means it is regulated by our body. It is not sudden, and it is like pre-planned. Like this cell has to die, and there are different processes by which these cells can undergo this death process. So another type of regulated cell death is this pyroptosis, where uh, this pyroptosis is occurring typically by this macrophages, and it is induced by inflammasome activation. So pyroptosis can contribute to pathogen elimination in different kinds of ways. Like first is the death of the infected macrophages prevents further spread of intracellular bacteria such as Salmonella and Listeria that replicate in these cells. This infected macrophage will die and will prevent the further spread of the intracellular bacteria and like Salmonella or Listeria that replicate in these cells. And second is it can also induce the secretion of the certain cytokines like IL-1 beta and IL-18, and it will release the mature form of these cytokines, and there will be a inflammasome mediated caspase 1 or caspase 11 cleavage from large precursors, and they will generate this inflammatory responses. So they can also induce the death of these cells, of these pathogen infected cells, by the this caspase mediated process, or they can directly uh, prevent the further cell by directly ingesting and killing this bacteria. So these are the two different types of regulated cell death that can contribute to pathogen elimination. So apart from that, there is, as I am already telling, that there are inflammatory processes which are happening inside the cell and which can directly kill the bacteria. So first is whenever there is a entry of the bacteria, it will activate the innate immune response in our body, and which will activate the phagocytosis by resident cells such as macrophage and dendritic cells. Like here we can see this is the bacteria which is breaching our epithelial layer, maybe the skin, maybe other mucosal surfaces, and it can enter. Like these are the first line of defense, like the anatomical barriers, which will prevent this bacteria or any kind of virus to enter. But whenever there is a breach in this system, then our innate immune system will be activated. And here you can see where this bacteria is getting entered into the cells, and whenever there is any tissue damage also, this there will be release of this chemoattractants, and these chemoattractants will induce other cells to come into the picture, like there will be local increase in the blood flow also, and there will be a capillary permeability also to help in the entry of these cells. And here, then what happens, there is a capillary permeability will increase, and this you can see through the blood flow. These are the RBCs, and here through the blood flow these cells are coming, and through a process known as leukocytosis, like uh, to the site of infection. Leukocytosis means these cells or the white blood cells which are known as the leukocytes will come into the picture. Here you can see first this neutrophil is coming, then getting attached to this blood vessels. These are the blood vessels, and attached, and then through a process known as extravasation, it is entering into this capillary space, means into the uh, sites of infection it is entering. So they the capillaries are becoming more permeable, and is all not only the cells it is allowing, but also it is allowing an influx of fluid into and cells inside this space. And then what happens, these neutrophils and other phagocytes will migrate to the site of this inflammation by a process known as chemotaxis, that these chemicals are getting attracted to the site of inflammation, and this will release certain enzymes and molecules which will then help in the destruction of this bacteria, either by phagocytosis, or it can by this enzyme it can directly kill the bacteria. So, as I already told, first neutrophil and then monocytes are coming to the site of infection, and there will be influx from the blood of the fluid by and there will be production of cytokines and chemokines and other mediators which will change in the vascular endothelial cells that will lead to an influx from the blood of the fluid. So this is one of the reason why whenever there is any pathogen which is entering in our body, and there will be redness, swelling, heat, pain that is occurring in our. So whenever there is a bite from the mosquito, we can see like there is redness in our skin and swelling of our skin. So this is nothing but because due to the accumulation of these cells and this accumulation of these cytokines, chemokines, and other enzymatic factors, which is like clearing the infection from that site. So whenever there is any kind of this redness, swelling, or pain in your body, you think it as like there is some pathogen which is trying, or there is a tissue damage which has occurred. So there is these molecules which are coming up together to remove the pathogen, or to like uh, repair the tissue. So this kind of local inflammatory response is happening in my body, and this is subsequently clearing of the infection. So any doubt is there? If any doubt is there, you can write in the text box also. You can unmute yourself also. Okay. So I am proceeding. So, as I already told, like there are different types of cells in lymphoid cell and myeloid cell, which I have discussed in the previous classes. And one of these cells, which is a innate lymphoid cell type, and uh, it is a natural killer cell. It is a type of a lymphocyte, and it is a it is also activated by infection, damage, or stress to enhance and regulate innate and inflammatory response. And collectively, these cells are known as innate lymphoid cells since they are lymphoid lineage. And natural killer cell was one of the first type of innate lymphoid cells which were discovered, and they have the unique function of killing cells that have become altered due to infection or stress. So this natural killer cell mediated cytotoxic activity is enhanced by IFN alpha production during early virus infection, an example of positive feedback regulation in innate immunity. So uh, in addition to releasing cytotoxic mediators, some activated NK cells also secrete cytokines and other pro-inflammatory cytokines like TNF alpha and IFN gamma. So this is one of the type of cells like natural killer cell which is also involved with some cytotoxic activity which will kill cells that has become altered due to infection or stress or even cancer producing cells like the cancer causing cells that also it can kill directly, and they only are known to produce this interferon alpha and different kinds of cytokines by which they mediate their function. Most like mostly this TNF alpha and IFN gamma. They can induce apoptosis of target cells if their activating receptors can recognize this infection or stress on the cells and can send stronger signals than their inhibitory receptors, which recognize markers of normal cells such as MHC proteins. So they can also induce the apoptosis of target cells. So uh, there are certain by their activating receptor means there are certain two types of receptors present, like one is activating receptor and one is inhibitory receptor. So activating receptor will activate the signal process from that cells, and inhibitory receptors is like will inhibit the signal from that cell. So why two types of receptors are present? Because if it is not possible that if some activating receptor is also present, there should be some inhibitory receptor, because prolonged activation of some receptor is also not good. It can lead to inflammation and other stress, and that signal needs to be stopped after a certain time whenever there is a clearance of this pathogen or clearance of the stress from the body. That signal should also be stopped by the inhibitory receptors. So two types of receptors are mostly present in many cells, activating as well as inhibitory. And whenever there is any kind of stress or infection, these NK cells will have their activating receptors which will recognize this stress and can uh, send stronger signals than their inhibitory receptors and can recognize markers of normal cells such as MHC proteins. So basically, these natural killer cells are having some certain cytotoxic activity. So apart from these natural killer cells, there are different other kinds of like innate lymphoid cells which also have uh, other cytotoxic activity. For example, these ILCs are assigned to one of the three groups, ILC1, ILC2, or ILC3, based on the cytokines they produce. Group 1 ILC consists of these NK cells as well as ILC1 cells. And here you can see these NK cells produce this IF and gamma TNF perforin granzymes, which will function as give immunity to viruses and intracellular pathogens, tumor surveillance, including cytotoxicity. And ILC1 cells, they will produce IF and gamma TNF. They also will give immunity to different kinds of pathogens like virus, bacteria, and parasites. Then group 2 ILCs will secrete IL-4, IL-5, IL-9, IL-13, and they will give immunity to helminths or wound healing. They have wound healing properties. The group 3 will contain different like LTI cells, like ILC-17 cells, ILC-22 cells, and they will uh, produce different kinds of cytokines. Like LTI will produce this LT alpha, LT beta, IL-17A, IL-22, and they are mainly involved in lymphoid tissue development, intestinal homeostasis, immunity to extracellular bacteria, etc. And ILC-17, due to the production of IL-17, they are named like ILC-17 cells. They will also produce IFN gamma, and they will produce immunity to extracellular bacteria. And ILC-22 will produce IL-22, and they also give immunity to extracellular bacteria, as well as they cause homeostasis of epithelia. So this, except for some NK cells and ILC3 cells, most ILCs do not have PRAS and not activated by pathogens. So they are instead activated by like cytokines and other mediators produced by local epithelial cells, macrophage and dendritic cells after PRAS stimulation by PAMs. So not every ILCs have these PRRs except NK cells, here NK cells and ILC3 cells. So others are having other receptors or instead are activated by cytokines and other molecules, and they will help in the clearance of the pathogens. So different kinds of populations of these ILCs are also there, which will secrete these different kinds of cytokines and will help in immunity to different kinds of pathogens. So, however, this I am talking about the activation of the innate immune system by different pathogens, but regulation of this innate immune response is very much important as well. Like there should, it has been seen like even if there is a mutation in one of the component, it can component of the innate immune system or the inflammatory responses, then there can be a there can be diseases which can occur due to the mutation in the components of this system. So here you can see this, some 60 children had a mutation in MYD88 or IRAK4 deficiency. So this I have already discussed in the previous class. Like this MYD88 is an adapter and IRAK4 is a protein, which is like all these two these two proteins are very much needed for the downstream signaling by different kinds of TLRs and other TRRs. And you what was seen that that 60 children had some deficiency in these two types of proteins, and these children were severely affected by bacterial infection, and many children in this many children, their the rate of mortality is also like seen. This is mostly, I think the data is from US. And so here you in A you can see like there is a decrease in the percentage of children with these deficiencies who are asymptomatic, reveals the incidence of the first severe bacterial infection during the first 50 months of life. So here you can see like patients without infection, like as the months is increasing, patients without infection, there is declining in the percentage of children with these deficiencies. So you can understand like how much important this all the components of this innate immune system is, and they can also lead to mortality if there is any kind of mutation. So here it is a survival curve of children with deficiencies of these persons who, here you can see like there is a with the increase in years like 20 to 30 years, it has become saturated, but patient surviving is uh, gradually decreasing. But here, after 5 years of age, you can see like there is no change. Like mortal that surviving percentage has become saturated. But before that, there is a decline in the survival rate of these children who are having these deficiencies. So mutation in this any kind of thing we can lead to different kinds of disease and can induce the mortality increase the mortality rate among the people, especially during the first initial years of the age where uh, like there will be highly severe infection. They will be infected with a high rate of bacteria, or there will be a severe infection in this children, especially because their immune system is not as developed as the adults. So there is an increased chance of mortality if there is any kind of mutation in this signaling system. So uh, there are genetic defects reducing the production and antibody effects of the IFN alpha and beta. So here you can see these red proteins, and these red proteins, when mutated, had different kinds of genetic defects in people in which these red proteins were mutated or having some mutations. So these viruses are taken up by these dendritic cells by specific receptors and are detected by specific TLRs. Here you can see different kinds of TLRs are present in the endosome, and this transport to the endosomes of TLRs 3, 7, 8, and 19 is dependent on the ER protein UNC93B. So UNC93B is a important protein, and which is important for the transport of these TLRs to the endosome. And it has been seen that mutation in this mm, different kinds of proteins. Like here, we can see like cytoplasmic signaling components activate transcription factors, including IRFs and NF-kappa B, leading to the synthesis of IFN alpha and beta. In humans, TLR3, UNC93B, TRAF3, NEMO, and IRF7 deficiencies are associated with impaired IFN production in response to certain viruses. So, people who have deficiency in these proteins like TLRs, 3 protein, TRAF3 protein, then IRF7 protein, NEMO protein, they have shown like there is impaired production of interferons in response to viruses compared to the people who have no deficiency in these proteins. So here also this bind whenever there is production of this IFN alpha and beta, this IFN alpha and beta will go and bind to this IFNAR receptors, as I already told, and there will be subsequent signaling. The JAK signaling will be activated, and it will lead to the production of different kinds of cytokines and other molecules, and also the transcription of the different kinds of ISG. So if there is a deficiency in this tyrosine kinase or STAT1, it has been seen that they are associated with different impaired IFN responses. So genetic defects, if is present in these kind of different kinds of proteins, these red are the mutated ones. So if this here, suppose in UNC93B there is a mutation, so what will happen? It will not lead to the transport of these TLRs into the endosome, and there will be no signaling. Similarly, if there is any kind of mutation in this IRF protein, so it will not lead to the production of this interferons, and there will be no activation of signaling in the virus infected cell in this interferon pathway, and there will be no killing of this virus infected cell, so that person will be more susceptible to different kinds of infection. So it can lead to different types of disease. Uh, So as I already told, like too much activation of our signaling pathway is not good. Like it can also be harmful. So not always the innate immune response or the inflammatory response which are produced by your cell is good, and it can lead to different diseases also. So uh, it should not cause any tissue damage in our, like too much increase in the production of cytokines or chemokines or other mediators should will cause tissue damage. So innate and for keeping us healthy, innate and inflammatory responses should use their destructive mechanism to eliminate pathogens and other harmful substances quickly and efficiently without causing tissue damage or inhibiting the normal functioning of the body system. So it should be, our system should activate our immune system in such a way that there should not be any kind of tissue damage, or not it it won't disrupt any normal functioning of the body system. Otherwise it can lead to different kinds of diseases. So one of the condition uh, which can be harmful to us is sepsis, and most dangerous of this condition is sepsis, which is a systemic response to infection that includes fever, elevated heartbeat and breathing rate, low blood pressure, compromised organ function due to circulatory defect. So our, if there is a too much response in our body, systemic response in our body in response to any kind of infection, it might lead to one of the deadliest disease which is known as sepsis, and there will be different kinds of symptoms, and um, this can lead to even death also. So sepsis results from septicemia, that is infection of the blood, in particular to those involving gram-negative bacteria such as Salmonella and E. coli. Um, and infection of the blood with this organism can cause sepsis and the systemic expression of pro-inflammatory cytokines. And left unchecked, systemic inflammation leads to septic shock, a highly fatal condition. So if there is excessive production of these cytokines and other this inflammatory mediators, then it can lead to a fatal condition known as septic shock, and it will definitely lead to death. And this will be like you cannot if you do not control excessive production of this cytokine, and you are seeing like there are like high breathing rate, low blood pressure, compromised organ function, and there will be fever, heartbeat will be increased, and your body cannot control the this regular function, and it will come this and it will lead to a uh, like condition known as sepsis, and which will be too much deadly for a person. So as innate and inflammatory responses can be harmful as well as helpful, they are highly regulated by positive and negative feedback pathway that generally keep the responses at the appropriate level. As I told, like there are two kinds of receptors: activated receptor or inhibitory receptor. So whenever there is a need for activation of our immune system, these activated receptors will be activated, and they will produce signals which will activate the signaling process, and there are inhibitor receptors, and whenever this activation of the receptor is done, they will produce this inhibitory receptors will act, and there will be inhibitory signals which are produced, so that this signaling is stopped and our body is returned to the normal condition. So there are various positive as well as negative feedback pathways which are acting together to keep this response at check level and at the appropriate level, so that there will be not too much higher production or not too much lower production of these different inflammatory mediators. So too much or too less is also not good. It should be in a balance. So, as you already know, our body is constantly trying to fight the pathogens. The pathogens are also evolving day by day. Every day they are evolving also to evade our innate and inflammatory responses. So, there can be different ways by which they can evolve or evade our innate and inflammatory responses. One is they can avoid the detection by pathogen recognition receptors. These are the different kinds of example by which they have evolved themselves to bypass our immune system. For example, this proteobacteria has a flagellin. It has evolved in such a way that it is having a mutation that is prevents it from being recognized by TLR5. Then there is Helicobacter, Coxiella, and Legionella bacteria that have altered their LPS structure or condition that is not recognized by TLR4. Then there is HTLV1 virus P30 protein which inhibits the transcription and expression of TLR4. Then there are several viruses like influenza, vaccinia, Ebola which can encode proteins that bind to cytosolic viral double-stranded RNA and prevent it from binding and activating RLR. So these are the different ways why they have evolved themselves to avoid the detection by the pathogen recognition receptors. Otherwise, what they can do, they can block the PRR signaling pathways, preventing activation of responses. For example, vaccinia virus protein uh, A46R and several bacterial proteins have TIR domain. So when there is TIR domain, they will interact with the TIR of these receptors and they will block the PRR signaling pathways. Then they will block these adapters from binding to the TLRs. Then there are several viruses that block the TBK1 IKAPA activation of the IRF3 and IRF7 required for IFN production. Then there is West Nile virus, which is having this NS1 protein that inhibit the NF-kappa B and IRF transport into the nucleus. Then there is Yersinia bacteria, which will produce these YOP proteins that inhibit inflammasome activity. The YOP protein inhibits transcription of the IL-1 gene. So by the production of certain proteins, they will block the PRS signaling pathway, preventing the activation of responses. Then there are other types of uh, pathogen which will prevent the killing or the replication inhibition. For example, Listeria, which is a bacteria, will rupture the phagosome membrane and escape to the cytosol. It has got some properties by which it will rupture the phagosome membrane and will escape into the cytosol, preventing phagocytosis. Then this Mycobacterium tuberculosis, which is responsible for this tuberculosis, is also will block the phagosome fusion with lysosomes and inhibit phagosome acidification. Then again this tuberculosis and Staphylococcus aureus will produce protein that protect them from loss and this reactive nitrogen species. Then vaccinia virus encode a protein that binds to type 1 interferon and prevents them from binding to the IFN receptor. In Ebola virus blocks the antiviral effects of IFN by preventing the nuclear translocation of phospholipid STAT1. Then this hepatitis C virus protein NS3NS4A and vaccinia protein P3L bind protein kinase R or the PKR, which is I already taught today. It is an ISG and block IFN mediated inhibition of protein synthesis. Then this herpesivirus and poxvirus encodes versions of anti-inflammatory cytokine IL-10 that reduces the local inflammatory response and T cell activation. Then you can understand how much these pathogens are also clever that they are itself secreting or modifying themselves so they can evade our immune system. So there is a constant race between the pathogen and the host, like who will win, kind of. If the pathogen wins, then we will we will have the particular disease and it can lead to severe consequences. And if the host wins, then we can easily clear away the pathogen. So every day our host is also evolving as well as pathogen also evolving to counteract each other and to like and to inhibit the other response. So uh, then comes the interaction between the innate and the adaptive immune system. As I already told, if there is a clearance of the innate of the pathogen by the innate system, then it is fine. But always one is dependent on the other, and innate immune system actually activates our adaptive immune system. So one dendritic cell is one of the key cellular bridge between innate and adaptive immunity, because microbes which are bound to the surface of these dendritic cells via the pathogen recognition receptor are brought from the site of infection to the lymph nodes where this all these adaptive immune cells are present, the T cells or the B cells are present. So this when there is activation of the dendritic cell by the PAMs, they will stimulate the cell to mature so that it acquires the ability to initiate the activation of naive T cells that will become cytotoxic as well as helper T cells. And these naive T cells are on the lymph node. So dendritic cells is bringing these microbes to the lymph node where they are being recognized by these B and T cells, and which are then getting activated and getting matured or differentiated into cytotoxics and helper T cells. Then various pathogens bind distinct PRRs on dendritic cells and activate signaling pathways that control what cytokines the dendritic cells will secrete. So this dendritic cell will activate certain signaling pathways and it will secrete many cytokines, and the cytokines will induce the differentiation of these T cells into different kinds of cells and will help in the mounting and immune response. Thus, the role of this dendritic cell is to ensure that, what type of adaptive response will be generated and will be effective against the invading pathogen. So, uh, this innate immune response is majorly involved in activation of the adaptive immune response through various ways. So, I don't know whether you guys have heard about this adjuvants or not, but adjuvants are nothing but they activate the innate immune response that increase the effectiveness of immunization. So there are certain types of uh, molecules or components which will help in the enhancement of the immune response in both laboratory animals and humans, containing ligands for TLRs or other PRRs. So these are known as adjuvants, and these will increase the immune response. For example, complete Freud's adjuvant is one of the most common kind of adjuvant used for immunization in experimental animals. Is a combination of mineral oil and killed mycobacteria. So, suppose when you are getting vaccinated, so it might happen that whenever you are giving a dead pathogen or an inactivated pathogen into your body, your immune system will throw away that. Like it will not accept it as a like it will not accept it and it will try to remove it. So there will not be any kind of um, to say activation of the immune system and there will be no proper vaccination. So for that, for vaccination, there is use of this adjuvant will increase this immune response so that this for example here with killed mycobacteria a combination of mineral oil is given so that which will uh, uh, like help this microbacteria to go and will increase this innate immune response inside the body. Another one is alum, which is a precipitate of aluminum hydroxide and aluminum phosphate, is used as an adjuvant in in some human vaccines. It has recently been shown to activate inflammasome, thereby enhancing IL-1 beta and IL-18 secretion and promoting inflammatory processes that enhance adaptive immune responses. Immunization with alum usually leads to the activation of T cells to become T2 cells, which enhance antibody responses. So modern vaccines, the COVID vaccine which we recently took, also use these adjuvants to increase the our immune response to that killed pathogens. And so modern vaccines make use of the connection between innate and adaptive immune response by including in the vaccine adjuvants substances that have been shown to activate the innate immune response and enhance the adaptive immune response. So, in us, means the humans, we are all studying this innate immunity, adaptive immunity with respect to us, like reference is humans. But what about the other animals which are present? So, there are different kinds of uh, like other uh, animals also have this kind of response, and this innate immunity has been shown to be appeared early during the evolution of multicellular organisms. For example, some innate immune effector functions such as antimicrobial peptides are found in invertebrate as well as vertebrate animals, plants, and even to some in fungi. Innate immune defenses in plants include anatomical barriers as well as PRs that activate antimicrobial innate immune response, some of which are similar to those found in animals. So here you can see a table where there are the different taxonomic groups which are present here. You can see, and you can see what are the different types of immune response which are present in this uh, animals. So here you can see mammals all are present like innate immunity, adaptive immunity, then protective enzymes and enzyme cascades, phagocytosis, antimicrobial peptides, pattern recognition receptors, lymphocytes, everything is present. In birds also you can see everything is present. In reptiles also almost everything is present. So here you can see vertebrate animals from mammals to jawless fish. All these responsible responses are present. So not only humans but other uh, family also like this amphibian, reptiles, bony fish, everywhere this immune response is present. Maybe it is different from us, the humans, but all these innate immune, adaptive immune, everything is present. But however, when you go to the invertebrate animals, here in case of arthropods, as you can see, innate immunity is present, but no adaptive immunity is present, or as well as lymphocyte. Here in annelids or earthworm, the adaptive immunity is also not present. It is not known whether they produce any enzymes also, and lymphocytes also is not present. In this porifera, the sponges also similar thing. And in case of plants, this adaptive immunity is again not present, but they have these enzymes and enzyme signaling, but no phagocytosis is present and no lymphocyte is present. So you can see that how this innate immune response is so much ubiquitous, because in all the cases you can see there is a presence of innate immunity, though in some invertebrate animals, like this, all the invertebrate animals, this adaptive immunity is not present. Only vertebrate animals it is present like us. Mm. So this innate immunity is present in all the organism, whether it is plants, whether it is invertebrate animals, or whether it is vertebrate animals. So you can understand how ubiquitous in nature this innate immune signaling is, and it is not specific to us. So this signaling can be studied in all the different kinds of taxonomic groups. So innate invertebrate and vertebrate immune response show both similarity as well as differences. So vertebrates and invertebrates share some innate immune response mechanism, including phagocytosis and production of antimicrobial proteins and peptides such as defenses. Invertebrate immune responses are best understood in Drosophila melanogaster, where first this toll-like receptor signaling was understood. It was not found in humans first. First, it was found in Drosophila melanogaster, where this toll protein responds to pathogens, where by activating pathways similar to those downstream of vertebrate TLR were known, leading to the production of antimicrobial peptides. And also, the invertebrates also use innate immune strategies not found in vertebrates, such as one is melanization. They change their what to say their uh, colors, and they protect themselves, which is not found in us, obviously. So these are the different aspects of uh, innate immune system, and as you can understand, it is broadly present in from invertebrates to vertebrates, though their signaling or the different mechanisms will be different. So today I am done with this lecture. So if you are having any doubt, you can unmute and tell. Okay, so no doubt is there. I hope you have understood everything, and if there is any doubt, you can reach to the portal where you can share your doubts, and or also you can email me. So thank you. That is all for today.